SARS-CoV-2 variants an update. Um, so what we are witnessing is a pandemic that is almost devastating the entire global human community. The SARS-CoV-2 is a highly transmissible RNA virus, which is implicated with COVID-19 disease, which was reported in Wuhan province of China in 2019. So the disease manifests within 2 to 14 days, and the symptoms are shared with influenza virus. So there is no pathognomonic symptom that is unique to SARS-CoV-2 infection, but it's rather found across any of the respiratory tract infections caused predominantly by viruses. And if left unattended, the infection can compromise the respiratory capacity, respiratory functions, leading to pneumonia, cardiac injury, sepsis, and multi-system organ failure. And the disease is transmitted by transmitted across humans, and the global dissemination of this disease is attributed to person-to-person contact and failure to use masks, or improper adherence to sanitary measures, and also due to limited accessibility to vaccines in some low-income countries. So this is what is the current scenario. As you will see that COVID-19 was declared a global epidemic by the WHO in March 2020. You will see that this is the current scenario. So the scenario is on 13th of December 2022. You will see that over 66 lakh individuals have succumbed to SARS-CoV-2 infection, and over 13 billion doses of vaccines have been administered. And you will see that the current scenario is devastating the global population, but Japan leads the table with 4,104 deaths in the last 28 days. And you will also see that 28-day deaths is 41,683 on 13th December 2022. But in the next slide, you will see that just in 15 days time, you will see that the 28-day deaths has drastically increased, 860,500 plus deaths globally. And again, you will see that the death rate in Japan has exponentially increased from the December numbers. Okay, the numericals between December and January is significantly high in several other countries like Japan, South Korea, US, France, Brazil, and Germany. And this is a testimonial to the fact that the virus is again continuing to exert its negative influence on the global population. So it is continuing to wreak havoc on the global population. So briefly about coronaviruses. Coronaviruses are distributed among animal species, domestic as well as wild animal species, especially wild animal species. And you will see that the viruses are widely disseminated among bat species. You have Microchiroptera, Megachiroptera bat species which harbor many of these viruses across the Sarbico virus groups, which are predominantly found among bats. And you will see all the other coronaviruses belonging to the Verbeco viruses, Imbico, Norbeco, and Quebeco virus. All these are different coronavirus species, widely distributed across different animal species in the wild, and also among certain domestic animal species. So the virus appears to have jumped into the human compartment through intermediate reservoir hosts, so malign pangolins and some reptile species appears to have harbored the virus before the virus probably might have jumped into the human compartment. So you will see that interhuman transmission occurs through the small droplets, larger droplets, direct contact with contaminated surfaces, very well established modes of transmission. But the transmission modes by laboratory exposure, blood transfusion, organ transplantation, transplacental route, or perinatal transmission remain ambiguous. There is not much of information, there is not much of evidence to show that the virus can be transmitted through any of these modes. But still, there can be speculations that virus can still be transmitted by these modes. The virus is a spherical virus with envelope. It's an enveloped virus with spike proteins. So there is a spike protein component which you will see here. The spike is made of two components, the S1 and the S2 component. The S1 component has knob-like structures. It's a trimer. You'll see three structures jetting out, and these are the components that are responsible for the virus to bind to the ACE2 receptors expressed on host cell. And the virus genome is about roughly about 30,000 nucleotides, and the virus has a single-stranded positive polarity RNA as its genome, which is of low stability, and therefore, due to its low stability, the virus is also prone for accumulation of several mutations. And with about 9.8 into 10 to the power 4 substitutions per site occurring yearly, you will see that the viral genome has open reading, 6 open reading frames. So some functional open reading frames. There are 6 functional open reading frames, and you have surface genes encoding surface proteins like the spike protein, envelope protein, membrane proteins, and also the nucleocapsid protein. So these are the surface proteins, the genes that contribute, that encodes for surface proteins of the virus. And functional open reading frames, there are about 6, roughly about 6. And you will see that the virus binds to the ACE2 proteins expressed on olfactory epithelium and also the type 2 pneumocytes especially. Of course, the ACE2 protein is found on almost all cells in our body, but they are more predominantly expressed on olfactory epithelial cells and also the type 2 alveolocytes in the alveolar compartment of the lung. So you will see that the virus binds with the help of its S1. The spike protein is made of 2 components, the S1 component and the S2 component. You will see that this is the virus, and we have the host cell plasma membrane expressing the ACE2. You will see that the S1 protein has 3 domains. So one of these domains is the apical part of these domains. All these S1 domain has receptor binding domain. You call it as RBD. This RBD is made of receptor binding motifs. There are motifs, several motifs, which actually make up the receptor binding domain to which the ACE2 protein can bind to efficiently. And this binding is something that is going to decide whether the virus has to infect the particular host cell. And what happens during the initial phases of binding, initial phases of infection, is that the S1 protein upon binding to the ACE2 protein will be acted upon by transmembrane serine proteases. One of those proteases is TMPRSS2, and also there is another protease which is called as furin. This furin can also act on the spike protein. So what happens here is the TMPRSS2 and the furin can act on the interchain disulfide bond that links S1 and the S2, and now what happens is this cleavage is going to bud out, is going to exclude the S1 from further downstream action with the cell membrane. So what happens is after binding, the cleavage happens, and this cleavage is going to scale off the S1, chop off the S1 component, where, I mean, exposing the S2 protein, S2 domain of this spike protein. This S2 protein is going to fuse the virus with the host cell membrane, forming a hairpin loop. There is a hairpin loop that is formed. This mechanism is almost the same as what normally happens in the case of HIV, where you will see that the V3 loop is getting exposed for it to bind to the chemokine co-receptors, the CXCR4 and the CCR5. Similar kind of a mechanism operates here. So this S2 is going to form a hairpin loop, paving way for the viral phospholipid viral envelope to bind with the the host cell membrane, allowing the virus to fuse with the host cell membrane, paving way for the virus to inject its single-stranded RNA into the virus, into the host cell. So you will see that the virus binds to the membrane, I mean, to the host cell membrane, and virus inject its single-stranded RNA. You will see that the RNA is recognized by the host cell ribosome for it to be translate proteins from these. This can serve as an mRNA, because being a positive polarity RNA, it can directly synthesize proteins. And then you will see that a polyprotein is getting synthesized, and this polyprotein is going to be acted upon by proteases, host cell proteases, and these proteases are going to clear, chop off these polyprotein for it to make smaller proteins. And you will also see that the positive polarity RNA can serve as a guiding RNA, and this guiding RNA can give rise to many other RNA components by transcription, and then these RNA can serve as a template for more single guide RNAs for it to become incorporated with the within the virus that is getting synthesized. You will also see that the viral components getting synthesized this way by translation can be can be used will be exploited will be taken up by the endoplasmic reticulum intermediate complex for it to synthesize the virus components, for it to pack the virus and to exocytose, for it to infect other cells in the closer vicinity. So the host pathogen interactions have already been discussed. So briefly, what happens is the mechanism has very well been explained in one of my previous presentations. So what happens here is there is influence of multi-system organ failure following infection by the SARS-CoV-2 virus. So coming to the role played by virus in undergoing mutations. So what actually happens here is we have a situation where viruses are distributed widely in the environment, and this environment has many hosts, many plant species, animal species. And there are many other different living organisms, living structures in the environment, which become exposed to infectious viruses, and this leads to exposure. Exposure can facilitate, can pave way for infection. So not that every living organism coming in contact with virus is getting infected, but there are random collisions which might ensue in successful infection, and this is again dependent on host tropism. Not plant viruses are not going to infect humans. Rather, plant viruses can infect only plant species. Animal viruses can infect only animal species. Human viruses can infect only human species. Likewise, host tropism again play a very crucial role in infection. And then the infection is getting spread across that particular population, be it plant species or be it animal species. In the case of SARS-CoV-2, or coronaviruses. It's going to be animal species, and also which live in closer harmony with humans in the environment. And then the virus is getting spread in the community, and then the virus has to adapt itself. After a virus has jumped from an animal species into the human compartment or into another animal species or whatever, it has to adapt to the conditions. But how is it going to adapt to the conditions? By necessitating nucleotide substitutions, deletion mutations, insertion mutations. So all these components, molecular components, are going to play a role in necessitating adaptation of the virus or the infecting pathogen, the evolving variant or whatever in the particular community. There is a recombination events, reassortment, gene reassortment events, selection events, and some components are going to select the virus to reside in the particular population for a longer duration. Some viruses, they are unable to necessitate nucleotide substitutions or these mutations, which cannot prevail in the population for a longer duration, will eventually disappear, which is what has been seen in the case of Delta variant, which is now, you will see that the Delta variant is not seen in the population, probably because it doesn't it is unable to undergo mutations, nucleotide substitutions, and because of this, the Delta variant is now has disappeared. It has largely been superseded by the other variants, the Omicron variants. And this is the actual picture, the big picture of what actually happens with a variant continuum, how variants appear in a particular animal species or in a particular community. So what is a variant? A variant is a virus. In this scenario, it's a virus. So variant virus has a deviant mutation or a complicated combinations of deviations, be it substitutions or be it deletions or additions or whatever. So all these can make up a variant, and this variant can have a markedly different phenotypic behavior, which you also can call this as a strain. That is the difference between a strain and a variant. A strain can have phenotypic changes, which can influence its behavioral aspects in the community, and therefore mutants are different from slightly different from variants and also strains. So what happens in a variant? What are the features that can attribute to the characteristic behavior of a variant? The transmissibility. The variant can be is now has now acquired a new set of phenotypic features, acquired a new set of phenotypic features due to insertion of a nucleotide or deletion of a nucleotide, and therefore it can be highly transmissible. It can be having a differential binding affinity. It can bind with a greater affinity compared to its wild variants. I mean, wild original virus strain, and this binding affinity can help its sustenance in the new environment. It can alter the disease presentation, the disease symptoms. So what we have seen is with the Delta variant is we experience anosmia and ageusia, but what we are seeing at the moment is there is no such kind of symptoms. What we have seen in the Wuhan variant, Wuhan strain, the wild strain was rhinorrhea, the running nose, but and also sneezing. And now what we are seeing is there is no sneezing. It has largely been silenced by these variants. So the disease presentation can be altered by a variant, and the pathogenicity, the ability of a virus to cause severe disease is what is called as pathogenicity. So pathogenicity attributes, the disease presentation attributes, can be altered, and the disease severity. You might have seen that Delta variant was causing severe disease and leading to high rates of ICU admissions and hospitalizations, which was not seen in the case of the previous the alpha variant and the beta variant, and also the Omicron variant. It has become less severe. So this is all, these are all some of the features. These are all the features that define what implications mutations might potentially have on variants that evolve from the parent strains of virus. So you will see here the variant. This is a computational image derived, which explains the spike protein of the wild strain of SARS-CoV-2, and you will see that there have been many mutations having happened over the period, which defined the alpha variant. You have different mutations in different genes which encode for the different domains present on the spike protein of SARS-CoV-2, which tells the alpha variant. So similarly, you will also see that the other variants, the beta variant, the delta variant, the gamma variant, the kappa variants, all these viruses having notable mutation, conformational changes attributing to their changes in any of these characteristic features. So, having said that, SARS-CoV-2 can cause COVID-19 severe or mild or whatever. Having said that, the virus can be contagious even without showing any symptoms of respiratory disease. The virus, the mutations, any of these mutations within the viral protein can lead to formation of variants, and therefore preventive measures must be attempted to neutralize, attempted to block the spike protein, the receptor binding domain, receptor binding motifs by any of these mechanisms, by using drugs, or by using peptides, or by using nanodecoys or nanosponges to prevent the virus from binding to H2. So we must design strategies to intercept the SARS-CoV-2 virus, not allowing the virus to necessitate the infection process. So this should be the strategy, because this is what is the attribute that is also available with use of neutralizing antibodies and so on. So the end result is to block the virus from binding to its cognate receptor, the H2 receptor expressed on the host cells. So this is the viral continuum. So what is happening? What is happening over the months or over the years? So we have seen that this April 2020. The red, the orange one denotes the. This side, the left side denotes the number of cumulative COVID-related deaths, and the right, the blue one, tells the number of cases in millions. So you will see the number of cases exponentially increasing over the years. So you will see that in February 2021 there was a report of different. The alpha variant, the B1.1.7 is the alpha variant, and along the same time you will also see the beta variant emerging from South Africa. The first variant that was described was in the United Kingdom, which is the alpha variant, the B1.1.7, followed by the B1.351, which is the South African variant, and then the Brazil variant, the B1, was reported almost the same time in February, March 2021, and then came the other variants, the B1617.2, which is the Delta variant, which was reported in India. The first case of the Delta variant was reported in Maharashtra in India, and then that attributed to severe, transformation of this epidemic into a nasty, scenario in India. I think it was in May to September, when India saw high rates of mortality due to the Delta variant. And then you will also see that the B1.1.529, which is the Omicron variant, emerged and was reported from November 2021 onwards. And this is how the virus, the variant is updating itself. Primarily, this is a mechanism for a virus to sustain itself, to remain in the human compartment. If it doesn't acclimatize itself to the human compartment, the virus may have to virus may become extinct. So for the virus to remain in close harmony, live in close harmony with the whole human compartment, it has to entertain mutations. It has to go hand in hand with the human system for it to survive in the human compartment. And that is why that is the reason why the virus is now mutating itself. As, unfortunately, some of these mutations can lead to deaths, and which is what is being experienced right now, globally. So, like any other viruses, as I said, SARS-CoV-2 is adapting to their new human and animal systems, and it is continuing to evolve genetically. And this will have different characteristics from its ancestral strains. As I had said, there are several factors which explain virus evolution. There are some selective pressures exerted by the host responses, which will subject the virus to mutate itself, to remain, to sustain itself, to maintain its association with the human compartment for over a prolonged duration. It has to, and then there are replication. Virus replication in other species can drive viral evolution. Prolonged viral existence in immunocompromised individuals, especially HIV infected individuals, can drive viral evolution. And also viral evolution can also happen because, especially in RNA viruses, because RNA dependent RNA polymerase, the replicase enzyme can interfere with virus proofreading functions, because you know that the virus has a very large genome, 30,000 nucleotide. It's a large virus, and therefore there are chances randomly which can lead to emergence of mutations. Some deletion mutations, some insertion mutations itself is sufficient enough to impact the virus behavior in terms of infectivity, transmissibility, and so on. So as you will see here, a paper that was published in Nature, I think it is Advanced Science by Wiley Publishers. So you will see that there are several mutations that have been explained. So one of these mutations, the first mutation that was reported in the Wuhan strain is one of those mutations, the N501Y. So you will see that the 501 is a location where exactly the mutation has occurred. So I think it's a substitution mutation. So this substitution mutation paves way for a change in the receptor binding domain of the spike protein, and this change in the receptor binding domain paves way for the virus to be taken up by immune cells, by endocytosis, or by any other cells, by means of endocytosis. And this endocytosis, easy makeover, easy, rapid endocytosis of this virus due to this change, due to this expression of this substitution mutation, leads to entry of the virus, easy ingress of the virus into the cellular cytoplasm of the cell, paving way for it to multiply in enormous numbers, and that way, by this means, the virus can proliferate in enormous numbers within that particular cell. And then it also provides an opportunity for the virus to infect many other cells in the closer vicinity. And what the point that I'm trying to tell here is, this mutation has led to high binding affinity of the virus, the receptor binding motif of the receptor binding domain of the virus, the RBD of the spike protein, to fuse with the paving way for easy fusion with the cell membrane. And then there was another mutation which has also happened roughly almost at the same stage, the HV 70, HV 69 and HV 70, which you can see here, the V70 deletion mutation. So it's a deletion mutation. So this deletion mutation appears to have a role in changing the S1 protein. The S1 protein, will express different amino acid, compared to the wild strain. And now what happens? Whatever antibodies that are produced against the wild strain will not be able to bind to this S1 protein, and therefore the virus may not be neutralized. So this is again another mutation that that was explained again in the alpha variant, the beta variant, and also in the gamma variant. You will see all these two mutations occurring across all the three viral variants. And then there was another mutation, which is the P618H mutation, which appears to play a role in enhancing the activity of purine and TMPRSS2. So by enhancing the purine activity and by enhancing the TMPRSS2 activity, the virus can get its the S1 S2 cleavage happening rapidly, paving way for the virus to infect the cell at a rapid pace. So this also has been explained in one of these in these variants. And we are also witnessing the B1617.2, which is the Delta variant, showing many of these mutations. And you will see the Omicron showing high levels of mutation. The blue ones, the baby blue ones. So all these are indicative of mutation. So as and when you see the down, see down the line, downstream, you will see that the number of mutations have exponentially increased. As and when you pass yourself from your eyes from alpha, beta across to delta, the Omicron variant, you will see the number of mutations have exponentially increased in the Omicron variant. All this attributed to deletion mutations, substitution mutations, and also and these mutations paving way for the virus to make itself fit to necessitate the infection phenomenon. So the first mutation that happened was the first ever mutation that happened in the D614. The D614 is the wild Wuhan variant, Wuhan virus. And in this virus experienced a mutation in the G mutation. Okay, so you will see that the D614G is the first mutation that happened. The G mutation is the first thing happened in the D wild variant, and that is the reason why you designate this as D614. So D614 is the wild strain, and the G614 is the mutant. The first mutant. Means the alpha variant. Okay. So this was reported, as I have said, in Southern Europe, which is the UK variant, and this has spread rapidly, and this became the most prevalent global genotype at least by 2020. Almost about 74% of all the published sequences were explaining the prevalence of this mutant strains. So we recently conducted an investigation in Indian population. So we have found that this mutation, that's D614G, was found across all the variants, the Delta variant, and also the Omicron BA1.1, BA2 variant, the BA1 variant. So all these having showing high level of viral fitness. They are highly fit to cause an infection. They are highly transmissible. They can proliferate rapidly. So this is the characteristic feature of the D614G mutation. So which has been shown also by our team. So the virus can be highly infectious. The studies suggest that the G614 mutation has led to high levels of infectivity, they are highly sensitive to neutralization. And then you will see that the original Wuhan strain had the RBD. The RBD was totally closed. Okay, the RBD domain was totally concealed. But what you will see in the G614, the alpha variant and the other variants that came downstream, you will see that the RBD was erect. The RBD was totally exposed outside, for it to infect, for it to bind to the ACE2 HAC ACE2 the ACE2 receptor with a stronger affinity. So what you will see is that the mutants, all these mutants are having exposed very well exposed RBD motifs, receptor binding motifs. And this has been very well been shown. It's one of the premier publications which has been published in Nature by Plante. So they have experimentally shown that this the D614G mutation, the spike mutation, alters the fitness of this virus, and this has been experimentally shown by the same group. Of course, this was a commentary paper that explains the viral fitness of the D614G that was published in the New England Journal of Medicine. So you will see that they compared the potential of the wild Wuhan strain and also the first mutant strain, the alpha variant. You will see that the alpha variant was able to, the first variant was able to replicate more efficiently than the Wuhan strain. So they cultured the virus on the human lung epithelial cell line, and they found that the G614 was more efficient in replicating in the lung epithelial cell line compared to the D614. It tells this is a testimonial to the claim that G614 is more binding with a more higher affinity, fusing with the host cell membrane with high efficiency, and that attributes to its proliferative potential within the permissive cell, and therefore this also explains why viral loads are very high in variants compared to the wild strain. And then they also tested this in human airway model. They simulated an airway model in vitro, and they conducted the same experiment in this model, and they again found that G614 replicated more efficiently, and then G614 really outcompeted the original Wuhan strain when both these viruses were introduced into human airway models. And then experimental investigations conducted on hamster models, to check their fitness. They intranasally inoculated both these viruses into the nares of hamsters, and then they found they also have a co-infection group, and they found that these viruses were also replication. The virus was also highly fit to replicate itself in high titers. So they again, the G614 outcompeted the D614, and therefore the virus is again was found to be highly fit. So virus fitness has also been proved in the case of variants. So and with the background information that age and comorbid conditions can serve as a factor for these. These were the two factors that we found were associated with contracting breakthrough infection. And having said that, vaccine-induced immunoglobulins lasts only for a shorter duration. We then came up with Omicron variant, which was explained in November 2021. So we also conducted some investigations. We analyzed some statistical. We analyzed the data with some high efficiency statistical tools, and we found that the Omicron variants, compared to the Delta variant, also was not, that was also highly competent to cause death. So we, especially the BA2 variant, you will see that the Delta variant, the B Omicron, the original variant, Omicron variant, and then the sublineages of the original Omicron variant, the BA1.1, the BA2 variant. So all these had the potential to cause hospitalizations, intensive admission into intensive care units, oxygen support, deaths. So this is again a testimonial to the fact that variants can cause havoc. And then the BA2 variant was we compared the BA2 variant with the Delta variant. We see that the BA variant was the time to death for the BA1 variant, especially the BA1 variant, was shorter. So what I would like to tell here is that although the Delta variant was causing high levels of the number of deaths was high in the Delta variant, the BA1.1 and the BA2 variant caused death, but within a shorter duration. The duration to death was shorter comparatively. The Delta variant, to kill a person, had to wait for a longer for a person to die due to Delta variant, and he had to remain. He has to be alive for a longer duration. But Delta variant really killed. Although the severity was less, it killed within a shorter duration. And then we also found that the Omicron variants were all escape mutants. They were the viral loads were high. So you can see here we have Delta variant and also the Omicron variant, the different Omicron variants. I could hear some background noise.
We request other participants to mute themselves, especially Rajan, Mr. Yeah, thank you, sir.
Some distractions. So sorry. Okay, so you will see that the Delta variant and we had Omicron, different other Omicron variants. So you will see that the viral loads were very high with Omicron variant compared to Delta. So you can see here this was also significant. Although the number of samples with Delta variant was lesser, you could still see that the viral load was relatively higher in the case of Omicron variants. And then, irrespective of whether individuals were vaccinated or, despite getting vaccinated, the levels of viral load was high in the case of Omicron, and this was not seen in the case of Delta. So, which tells that Omicron variants were largely escape mutants. So they had mechanisms. Omicron variants had mechanisms. They evolved mechanisms to evade from probably neutralizing antibodies, or even probably from escaping themselves from CD8 positive T cells or whatever. So this potentially could be strategy why the viral loads were high in the case of Omicron, opposite Delta variant, which still was susceptible to antibodies or CD8 T cell mediated decline in the number of viruses or Delta variant viruses. And then you will see that despite getting vaccinated with Covishield or Covaxin, you will see that the viral loads are high with Omicron variants, but not in the case of Delta variant. So the virus appears to have evolved, downstream from Delta variant, for it to, develop strategies, evolve strategies to escape from, armory of the host. So what happens at this moment in time in Tamil Nadu? This is the data that is about to be published. So this data is about to be published. It's in the submission stage. So you will see the dynamics, the updates, the updated version of cell phones. Whatever you see. So you will see that this was in January, um, uh, uh, you know, um, 2022. You will see that these numbers are all corresponding to the different Omicron variants. So you will see the BA1 was more predominant, and then there was also some BA2, 21%. And then in February, you will see that the BA2 was totally outnumbered the BA 1.1. BA 1.1 is now being as shrunken. You'll see here just 3% from 48%. And then over the next 2 months, you will see that the BA2 was predominant. You can see here in March and also in April, you'll see the BA2 was predominant. And then you will see that in May, you can see here the BA2 is now getting superseded by BA 2.38. This is the variant that was almost in par with the BA2. And and then you will see the BA5 just getting entry here. And in June, you will see that the BA5 has totally superseded the BA 2.38 and also the BA2. And you will see in July and also in August, the BA5 was predominant. And then the BA 2.75, which was also called, some people also called informally a Centaurus something. And you will see that the Centaurus variant is predominant in September with 1,072 cases reported. And then you will see the entry of XBB. So you will see that the XBB expanded in October, and you will see in November the XBB variant was very high, and it was totally superseding the other variants. And you will see that now in December, the XBB variant has been superseded by the BA 2.10.1. So this is how the dynamics, the continuum is underway. So Omicron are a predominant active variant in Tamil Nadu at the moment. So you will see 8 of the 8,901 Omicron cases reported with COVID-19. WGS genome sequencing available. You will see the Omicron variant occupying 98%. So 8,758 of the 8,901 COVID cases are because of Omicron. You will see there are only 74 Delta variants. So what I heard yesterday from Dr. Raju was that from the Directorate of Public Health is that Delta is again slowly catching up. Delta is slowly catching up. So in the last one week or so, there have been many cases of Delta. Delta is slowly catching up. I don't know whether Delta is going to re-emerge again, but you will see that Omicron is the predominant variant. And this is the skyscrapers across January to December. You will see the Omicron is the predominant variant, and slowly you will see Delta is slowly, jetting out. Of course, the others are not reported, but Omicron is the predominant. But I don't know what is going to happen in the future. So this is a report that I received from the Directorate of Public Health, a formal report. So as per the guidelines of the Government of India, airport surveillance has been intensified in all the 4 international airports in Chennai, Coimbatore, Trichy and Madurai. And international travelers, especially, alighting from China, Japan, South Korea and US flights are screened for COVID-19. All the COVID positive cases are referred to the Tamil Nadu State Public Health Laboratory in Chennai. And then of the 106 COVID positive samples reported, 98% of these samples have been sequenced, and 14 COVID cases have been detected from airport surveillance. This is the data that I received until day before yesterday. And so far, Fortunately, BF7 variant, which is actually wreaking havoc in the West, especially Japan and US, Brazil, have not been detected in the 6 samples which were, obtained from this surveillance, campaign. So that brings us to the end of this lecture session for today.